So in the last lecture segment, I introduced the categories of the macromolecules, also called the biomolecules. These are all organic compounds, meaning they have carbon as their in their skeletal structure. And the four categories you recall are carbohydrates, lipids, proteins, and nucleic acids. The carbohydrates, the proteins, and the nucleic acids are all groups that contain monomer units, which are smaller molecules. And then when those smaller molecules are joined together, like beads on a necklace, it creates a larger macromolecule, which is called the polymer. So the good news about those three categories is that once you learn to recognize those monomer structures and the names of the monomer structures, the polymers are simply multiples of those monomers joined together. So in general, all polymers and monomers, the way polymers are assembled from monomers and how a polymer is disassembled back into individual monomers is these two reaction types. So a dehydration synthesis reaction is defined as the reaction that joins monomers covalently to form those long chain polymers. And also, as a part of dehydration synthesis, not only is the polymer a product, but water is a product. The opposite reaction is called hydrolysis. This reaction disassembles a polymer into many monomers. So the polymer, the bonds that are holding those units together are broken, and the polymer reactant becomes a group of monomers as a product. And in this reaction, water is a reactant. And so you do need to know the definitions of these. There are some diagrams here, as you will see, and we will go through these. This is not my favorite diagram, but you tend to see this similar diagram like this in various places, so it's probably good to at least be familiar with it. In this picture here on part A, you have these two units that are already joined together, and they're just using this little ball to represent whatever monomer unit is relevant for the discussion. And there's two of them together, and there's another monomer here on this side. Let me get my pointer out. And I'm on picture A. I don't know if my pointer is showing up for you. And when that H that's sticking out, let me get a pen. When the H that's sticking out right here and this OH that's on the other piece come together, they form water, which leaves the molecule, and it creates a bond. So here you have a new bond here. The thing I don't like about this is they actually have two arrows happening, and that's not a standard way to do this. What would be standard is we have the reactants here. We have one arrow, and then here are our products. So really the water should be shown on this side as a product. So I don't really love the fact that they have a second arrow going off in a different direction. Similarly, over here, you have in this hydrolysis diagram, you have a polymer, three monomers that have already been joined together to make a polymer, and water is on the reactant side, and then you have an arrow, and then you have two pieces. You've broken off one of those monomer units in the process of the hydrolysis reaction. So dehydration synthesis is a building reaction. You're taking smaller components and joining them together to create something larger, a polymer. And hydrolysis is the opposite. It starts with a polymer, water is a reactant as well, and bonds are broken, and the products of the hydrolysis reaction are smaller components, monomers, or smaller polymers. So those are generally the names of the reactions that are going to come up again and again and again in this course. So I'll be asking you many times, what's the name of a reaction that joins monomers to form polymers or joins smaller molecules together to make larger molecules? The answer is always dehydration synthesis. What is the reaction that breaks down larger molecules into smaller molecules? The answer is going to be hydrolysis. Hydrolysis, by the way, is also known as digestion. So that's maybe a word that you're more familiar with. But we use the scientific term hydrolysis. So we're going to move into our first category, carbohydrates. Carbohydrates as a category has maybe a little more information in this chapter anyway than some of the other categories. But for each of the categories, we're going to get some familiarity with the monomer or maybe an example monomer, and you'll be familiar with that one. You're not going to be expected to know everything, but we're going to focus on like one example monomer. And then once we get used to that, we can then see what it looks like when many of those are joined together to form the polymer. So carbohydrates are good for energy storage, and they're also good for structure. So creating parts of the cell, like the cell wall that holds plant cells, nice and firm. Bacteria have a cell wall. So carbohydrates are not only good for energy, but they're also good for structure. The monomers in this category are called the monosaccharides. And by the way, the polymers are called polysaccharides. But the most important monosaccharide that we're going to really focus on as our example is called glucose. Glucose is a 6-carbon molecule. So there are 6 carbons in its structure. And it also has 12 hydrogens and 6 oxygens. And so in this diagram, let's see, where is glucose? Right here. Now this is showing you the way that chemists will draw the structure of an organic molecule. And what they're showing you here, if you look closely, do you see these little numbers? 1, 2, 3, 4, 5, 6. They're counting for you the carbons. But what's interesting is when you study organic chemistry, because it's understood that carbon is the skeleton, they usually don't draw the C every time it occurs. So when you see this little blue one right here, there's a carbon, but it's implied. Like you would know that it's there. Right here, there's a carbon. The number 3 right here. Number 4 carbon right here. Number 5. The number 6 carbon, though, is shown. So they can draw it. They can write the C, but they don't have to. And so the way that you know if there's a carbon there is if these lines, which are the covalent bonds, when they intersect at a point and they don't indicate a symbol for an atom there, then you're allowed to assume that's implied to be carbon. So there are 6 carbons in that ring, 6 carbons in that molecule. Carbons 1, 2, 3, 4, and 5 are in that sort of hexagon shape. And then the 6th carbon sticks up, I think of it as like a little flag sticking up above the ring. So we call that hexagon shape the ring structure because it's round-ish. And it's a hexagon. And so a lot of times in organic chemistry, sometimes we get so lazy that we just draw a hexagon when we mean glucose, because glucose is such an important monosaccharide and it's so common in organic chemistry and in biology that when somebody draws a hexagon and doesn't say anything, you can assume they mean glucose. If they're trying to show you something else, they would need to say so, because glucose would always be the assumption if somebody draws a little hexagon. All right, so you can see the structure there. You don't have to worry about the other structures that are on this slide. Some of them we'll come to later, but for right now, we want to focus on glucose, this 6-carbon hexagon-looking structure. And it's got 6 carbons, but it also has hydrogens, and you'll recognize the hydroxyl groups that are there as well, the OH. So if you have could be 2 glucoses or 2 of any single sugar, simple sugars, and they get joined together covalently by dehydration synthesis, which is what's being shown down here. Try to look, don't worry so much about every little detail, but can you see that this is a glucose here, and this is another little sugar here, and then the OH and the OH here are going to interact and form water. That's going to leave one oxygen behind, and on this side of the arrow is where your products are. So these two end up joined together, and that's the beginning. It's not a polymer yet, but we would call that a disaccharide, meaning a molecule that really contains two component sugars in it. And you can imagine if another sugar was then joined in covalently on the side, that would be a trisaccharide. We usually would go pretty quickly to the word polysaccharide. But this dehydration synthesis can happen over and over again to give us this long chain, and that's what a polysaccharide is. So individual sugars like glucose, little monosaccharides, your cells like to use those for energy. We'll learn more about that in chapter 7. When your cell joins together monosaccharides to make a polysaccharide, it's because it's storing the energy for later. It's kind of packaging it up to keep it for later. But also cells will join sugars together to build some of these structures that they need for the cell itself. So that's why I said that polysaccharides are used not only for storing energy, but also for structures. So polysaccharides are defined as just long chains of monosaccharides. So when you're looking at a picture, if you see little hexagons, even if they have little hydroxyls and things sticking off, kind of look for that hexagon in the amidst the structure, and you see a bunch of them linked together, and that's what you're looking at. That's a polysaccharide. So it's a long chain of monosaccharide units that are joined together by these dehydration synthesis reactions. In plants, they create polysaccharides called starch. And so that's a plant's molecule for storing energy. Animals, though, in their cells, they build a polysaccharide that's slightly different, and it's just a little different in shape, called glycogen. Also a polysaccharide made of glucose. Starch and glycogen are just glucose molecules, many of them, joined together in these long chains. And plants also build polysaccharides called cellulose, and that's made of glucose units joined together. And some animals make a structural polysaccharide called chitin. Chitin is not strictly made out of glucose, though it has some other components in it. But chitin would be the exoskeleton of an arthropod. An arthropod would be like an insect. So think of a June bug. In the spring, when those June bugs are everywhere and you step on them and they crunch, that's the chitin. That's that carbohydrate structure that protects their outer body. So it's a type of polysaccharide. And then in this picture, I give you some diagrams so you kind of see what they might look like. So this would be a single unit of glucose, a monosaccharide. This would be a very short chain of glucoses. So that could be called a polysaccharide, very short polysaccharide. And then here, if you kind of think about zooming out and you're looking from a distance, you can see if you look closely, this is a whole bunch of little hexagons joined together in a chain, and then it spirals. But you can still see it's hexagon, so that's a polysaccharide. This is a polysaccharide. Do not worry about the exact names. We're going to just classify. Monosaccharide, just one hexagon. More than two, we just say polysaccharide. So here, this is a monosaccharide, hexagon, one glucose, polysaccharide, and then zoom out, and this is a bigger polysaccharide here. It's actually 3 polysaccharide chains that are kind of wound around each other. So monosaccharide, glucose, polysaccharide, several or many glucoses hooked together in a chain, sometimes spiraling even. So we get to recognize what those look like. And we'll have some practice when you get to your post quizzes. I'll show a picture like a hexagon. It'll be a glucose, and I'll say, what is this? And you could either, on the answer sheet, on the answer choices, it'll either say glucose or it'll say monosaccharide, and just whichever one is there, you pick it. If there's a bunch of hexagons hooked together, whether they're spiraling or just straight chain, you say polysaccharide. I won't have you do anything more specific than that for that visual identification. So the next section is, the next category are the proteins. And proteins are the tools of the cell. They're arguably the most important molecule in the cell. And I hate to admit that because I'm a geneticist. I want DNA to be the most important. And certainly DNA is very important. But if I'm being honest, if I'm being fair, proteins do all the work. They are the number one most important biomolecule category. Not that a cell would be okay without the other categories, but proteins are definitely the main one. So proteins, they do everything. They catalyze chemical reactions. In various definitions, they are used for defense. They can be used for transport of materials, whether it's transport of things within a cell or transport of things outside the cell but in the body. They're used for support and structure. They hold things up. They make up some of the structures in the cell. They make up structures outside the cell in the body. Motion, regulation, storage. So there's so many things that proteins do. So in other words, I wrote this summary. In other words, proteins pretty much do blank in the cell and in the organism. And the blank is everything. They do all the work. So the monomers of the proteins are called amino acids. And the main structure of the amino acid that you would need to be able to recognize for the post quiz and the exam eventually is shown here. So it's not a hexagon. And what you notice immediately, I hope, is that there is an amino functional group and a carboxyl functional group in the amino acid. So they're showing the amino functional group as NH2 kind of turned around in a backwards way, but you remember it's the only group that has nitrogen. Over here, the carboxyl group, you remember that the carboxyl group is a carbon with a double bond to oxygen, and then also a bond to an OH, hydroxyl, so that's carboxyl. And sometimes when we're being lazy, we just write COOH. So when you see those, that's the key that that's the amino acid. And then there's always a carbon in between those two groups. There's always a hydrogen attached to that carbon. And then there's something called the variable group, or the R group, or the side chain. Sometimes it's called the side chain or the variable group. Now the side chain, there are 20 possible little side chains that are very commonly found on amino acids. So 20 different R groups that we tend to find on the amino acids that we have found on Earth. So each amino acid is the monomer. That's the monomer of this category. And then what happens is when the amino acids are joined together in a long chain, then that is called a polypeptide or protein. So a polypeptide is an unbranched chain of amino acids. There aren't any branched chains of amino acids. They're always these straight chains. And a little bit like some of the carbohydrates, sometimes they make spiral shapes when you have a really large polypeptide chain. But just like the polysaccharides are made by dehydration synthesis, joining together the monosaccharides, in the same way, polypeptides are made by dehydration synthesis with multiple amino acids being joined together. So, the dehydration synthesis is going to come up again and again. It's always going to be the building reaction for the polymer version. So in the protein category, you have amino acids. Here's one here, and here's one here. And then a reaction happens that makes water. And then also what's created is a larger molecule. And when you have two amino acids together and that's all you have, you could call that a dipeptide. But again, pretty quickly, just like with a disaccharide, pretty quickly, if you add a third one and a fourth one, we just immediately kind of go to the word polypeptide. So I'm going to show you what the R groups are. This is not from your textbook, but they're highlighted here in yellow. You do not need to memorize them. But these are the R groups, also called the side chains or the variable chains. And so every amino acid has an amino group and a carboxyl group, but it also has one of these 20 R groups. So there's 20 different monomers, slightly different monomers in the protein category. Now, just like with the carbohydrates that I said, sometimes they form straight chains and sometimes they form spirals. Proteins aren't any different. The shape of the protein determines its function. That was a fundamental rule from chapter 1 of everything in biology. So shape and structure determines function. Proteins have pretty complex shapes, to be honest with you. And if you study protein biochemistry extensively, you will get to delve into all of the details of protein structures. We typically say that proteins have 4 levels of structure. Now, the first level is this: what of those amino acids are hooked together, because proteins have at any one position 20 different possible amino acids that can be hooked together with 20 other different possible amino acids. So you can have a lot of variation and slight variation in what a protein is actually made of in terms of its individual amino acid composition. But after those amino acids are joined together, some regions of the polypeptide will make a little spiral. That's called an alpha helix. Some parts of the protein will make a little, like a hairpin, like fold back and forth area. That's called a beta sheet. And ultimately, the whole polypeptide will fold together into a 3-dimensional shape. The overall 3-dimensional shape, though, of a protein is hard to describe with geometric terms. It's what I would call equivalent to the shape of, in many cases, the shape of popcorn. When you pop popcorn, it has a shape, that popped popcorn kernel. Each popcorn has a shape, but there's not really a word in geometry. It's a very irregular shape. There's parts that stick out and parts that are kind of little niches and crevices. And so what we call the third level, or the 3-dimensional shape of the protein, of the polypeptide, it's hard to have words to describe it, but every protein forms the shape that matches with its function. So different proteins, protein insulin will fold into the shape that matches how it functions in the body. But a different protein, like a digestive enzyme, will fold into a shape that allows it to do its function. So the shapes are all different, just kind of like the shapes of popcorn are different, but there's really not a good way when you're first learning this vocabulary to tell you. So we usually just use the term irregular and globular, but that doesn't quite satisfy needing to be able to express that irregular, unusual shapes. But they're very, they have little crevices and then little sticky outy sections and such. Now, there is another level of structure. After a polypeptide has folded into its 3-dimensional shape, there is another level that some polypeptides can reach and some don't need to, but it's called the quaternary structure. The 4th level is where multiple polypeptides, each folded into its own 3-dimensional shape, come together and attach to each other. And then they do a certain function in the body. So it's a very complicated system, and you will learn more about this if you go further in biology. But the main thing is that there is a three-dimensional shape to the protein, and the three-dimensional shape matches what the function of that protein is. So if a protein, for some reason, gets mangled up or loses its shape or loses its structure, then it will also lose its function. So proteins can be what we call denatured, unfolded, or disrupted in their shape. A good example of denaturation of proteins would be when you cook an egg white. An egg white is full of a protein called albumin. And when you first crack open the egg, it's slimy, and the egg white is very kind of watery and slimy. And in that case, the albumin, all the albumin in that egg white is fully folded into its three-dimensional shape. But when you cook it on the stove, what you're actually doing is you're breaking apart, unfolding. You're not breaking apart the amino acids, but you're unfolding the shape. You're stretching out the shape in a way that wasn't, that's not intended. And so you're denaturing the protein, and it changes. It goes from being slimy to being kind of solid, like an egg white omelet. And so that's denaturation. All right. So denaturation is caused by, and you can see the second one on the list, this is no particular order, temperature. High temperatures disrupt the structure and therefore denature the protein. Cold temperatures don't do that. If you freeze an egg white, it doesn't cook it. It doesn't turn white like an omelet. But high temperatures unfold the proteins. You can also, if you dropped, I don't think I would try this, but if you dropped an egg white into like hydrochloric acid or something very acidic, it will turn white and be like a cooked egg. But in that case, you haven't applied heat, you've applied acid. But acid and base will also denature a protein in the same way. And then salts, if you put too much salt into the environment of a protein, it will tend to have some unfolding effect on that protein. And then pressure. We probably don't have as much experience with those things, but temperature, I think that's why we cook any food, is to, if there's any bacteria or microorganisms, what we're actually doing is causing those bacteria, the proteins inside the bacteria that are doing all the work of that cell, we're causing them to be denatured, which is, for that bacterium, the definition of death. We're destroying the function of all its proteins. Therefore, that bacterium is dead by the time we eat that food. So we're using heat to kill microorganisms in our food so that they're not still alive when they come into our body. Because if you ingest live microorganisms, then you can get sick, food poisoning and such. So that is why we use high temperature cooking, but you can also do acid. So when you eat pickles, for example, anything pickled tends to, the acid of the pickling will kill any microorganisms that might be there. So it's good for storing food, which is what I have on the next slide. Food preservation and preparation focuses on denaturing the proteins of the microorganisms that might be in the food. And so the microorganisms spoil food because they're decomposers, but they can also make you sick. So they're harmful, and they can be parasites or they can just be pathogens. Parasite is something that stays inside of you and just lives off of you. Pathogen is something that makes you sick. I mean, you don't want either one of those things, right? So proteins perform all the functions of the cell and the organism, not only your cells, but also inside the microorganisms. So if food is contaminated with microorganisms or parasites or pathogens, if you eat those, you're putting them into your body. But if you denature the proteins, usually by cooking, but you can use acid or you could use other things, then you can then eat them, and you're just eating the dead organisms. But the dead organisms don't hurt you. So anyway, and you can't see them because they're microorganisms. So that is how that comes into play in your day-to-day life. Okay, there's 2 more categories. I'm going to take a break, and you might want to take a break as well. And we'll come back and look at nucleic acids, and we're going to look at lipids.